Case Western Reserve University's Institute for the Science of Origins proudly presents the Origins Science Scholars Program. The Institute advances the scientific understanding and application of origins and evolution of human and natural systems. The Origins Science Scholars Lectures are presented with the assistance of Case Western Reserve University's Siegel Lifelong Learning Program, College of Arts and Sciences, and Media Vision. Tonight, it's my great pleasure to introduce Johannes Haile Selassie, a world-renowned expert in early hominin evolution. Dr. Haile Selassie is curator and head of the Department of Physical Anthropology at the Cleveland Museum of Natural History and has discovered and elaborated on critical fossils in the human evolutionary record. Please join me in welcoming him.
It's always exciting to be here with you guys and talk about one of the subjects that I really like the most, which is human origins and evolution, fossils. Who doesn't like fossils? But today I'm going to tell you a little bit about the fossils that we've been finding from a site called Woranso-Mille, which is located in Ethiopia. And I'll show you where it's located momentarily. But I just want to say that Woranso-Mille has been one of the most important sites in recent years, particularly in terms of the fossils that are coming out of the site, and also the information that we're trying to extrapolate from the fossils that we're finding, and what new knowledge we're adding to our understanding of human origins and evolution. Now, before I start showing you any of the slides, I want to say that some of the field photos that you're going to see were shot by Dale Omori and John Mingles, and some of them, the good ones, are mine. So to start with the location of Woranso-Mille, it's located in Ethiopia, of course, and it's located in the Afar Rift of Ethiopia, and whenever you hear about East African Rift System, for those of you who were at Dr. Nengo's talk last week, he's explained about this rift formation and how far it goes to the south and also to the north. And I wouldn't be repeating that, but I just want to say that the East African Rift System is a very dynamic area where you have three different plates, pulling apart from each other, like in different directions, and creating this huge rift valley, which crosses Ethiopia and Kenya and Tanzania and goes all the way to Malawi and Mozambique. And a lot of times when I give talks on fossils, people ask, like, why this place? Why do you always find all these fossils in Eastern Africa? Why not somewhere else? And the answer is because of all these geological activities, all the tectonics that's happening in Eastern Africa, and the fact that it's opening this ancient deposits for us to go and look for fossils from the old times, that's one of the reasons why this area is really important. Not to mention that this is also located in the tropics, which is the favorite place for most primates. So when we talk about Eastern Africa, particularly the Horn of Africa, we have an area which is called the Afar Triangle. Now, this is probably the most productive area in terms of fossils, and people have been going to this area for the last almost 50 years now, since the discovery of Lucy about 45 years ago. And they've been collecting numerous fossils of not only early human ancestors, but also other animals that lived with them. So when you look at, this is like a two-dimensional view of that area that I showed you, and you see that there are so many sites within this Afar Triangle area, the most prominent of which are Middle Awash, Gona, Dikika, Hadar, Ledigararu, and now Woranso-Mille. So Woranso-Mille is like relatively recent in terms of its discovery. The other sites have been known for the last 40 years or 45 years in the case of Hadar. And each of these localities have been really significant in their own ways in terms of what kind of information they give us to understand our evolutionary history, our cultural evolution, our technological evolution, and so forth. And Middle Awash being one of the sites where we have the deepest set of deposits sampling the last 6 million years of our evolutionary history, most of the species that we know today in terms of early hominins, almost 90% of them are known from Middle Awash. And as you go further to the north, you have Gona, which is where we have the earliest stone tools at 2.6 million years ago. Hadar is the site of Lucy. Dikika, Selam's locality, Lucy's child, nicknamed Lucy's child. And further north, we now have new areas such as Lady Gararu. You might have heard that now the earliest specimen that is assigned to the genus Homo was found from Lady Gararu at 2.8 million years ago. Now that pushes the record of the genus Homo from about 2.4, 2.5 to 2.8 million years ago. And Woranso-Mille is now the site where you would go to find fossils that are dated to between 3.6 and 3.8 million years ago, because that time frame has not been sampled from the other localities that I just mentioned. So it's really important in its own way, particularly in addressing specific questions that we were not able to answer before the discovery of Woranso-Mille. So Woranso-Mille, as I said, is a place where you go to find fossils that you would otherwise not find other places like Middle Awash or Hadar or Gona or Dikika. But the record is so fascinating that in terms of the yield that we have from that specific region in the Afar Rift, all of these yellow dots represent some species or fossils of early human ancestor species that have been documented from that specific area. And the Woranso-Mille samples that time period between 3 and 4 million years ago, particularly 3.6 to 3.8 million years ago. So we started work at this site in 2005. The site was found in 2004, but we started like really serious investigation in 2005. And since then, I came back from the field like 3 weeks ago. And so far in the last 13 years, we've been able to collect about 11,600 specimens plus. And these specimens include representatives of hominins, of course, that are dated to between 3 and 4 million years ago. We have various species of monkeys, cercopithecids, bovids of all sorts, suids, carnivores, and a lot of other mammals like rhinos, giraffes, and hippopotamids, and so forth. So it's been really productive in terms of the number of fossils that we've collected so far. And as I said, we don't go out there just to collect early human ancestor fossils only, because if we do, then we're going to miss a lot of the information that goes with our understanding of human origins and evolution. So we collect everything that we can identify by element and what species it belongs to. But as at other places, the hominins have always been really rare. You don't find them by the thousands, okay? And if you look at most of these sites that we know of that have yielded human ancestor remains, the percentage is usually less than 1%. And at Woranso-Mille, it's not different from what we have from the other localities, but a little better, because here it's almost 2% based on the fossils that we have collected so far. But you also see some unique distribution here, particularly the monkeys. Cercopithecids are very, very abundant at Woranso-Mille. And that requires some kind of ecological explanation. Why do we have so many monkeys when they're really rare at other localities of similar age? So those are the kinds of things that you need to observe and try to understand the ecological factors behind the abundance of one group versus another. Why do we have more hominins, for example, compared to Laetoli, which is also an Australopithecus afrensis site? Why do we have more monkeys at Woranso-Mille than other places, like even in Kenya or other localities in Ethiopia? So there are so many questions that we would like to address based on just what we have collected. But today I'm just going to concentrate on the hominins, because that's the most exciting to me and to you, of course. So there are about 220 fossil specimens of early hominins that we have recovered from Woranso-Mille. I just want to show you some of the major elements that we have discovered from the site. and talk a little bit about why are these fossils really important. A jaw by itself may not tell you a lot. But when you have multiple things and you can actually start looking at variation between like 2 mandibles, 3 mandibles, you get more information than what you would from a single specimen. So when you ask that question, the answer is they really help us address some of the questions that we have in human evolutionary studies. Now, our questions in our human evolutionary studies are not limited to 1 or 2 questions. We have plenty of questions that we would like to answer. But based on the fossils that I have, I'm really concentrating on answering only a few questions. Especially if there is a hypothesis that has been proposed based on discoveries before Woranso-Mille, then the Woranso-Mille specimens might help test that hypothesis. And one of which is the Australopithecus hominins, Australopithecus afarensis chronospecies lineage. These are taxa that are considered as ancestor descendant. We have good fossil evidence from Australopithecus hominins dating back to between 3.9 and 4.2 million years ago. We also have good fossil record for Australopithecus afarensis that starts about 3.6 to 3 million years ago. Now that time period between 3.6 and 3.9 has always been empty in terms of fossils that have been recovered. So Woranso-Mille plays a major role in filling that gap because we have fossils dated to between 3.6 and 3.8 million years ago. So that's one thing that I would like to see with the fossils that we're finding from Woranso-Mille. The second thing is hominin diversity between 4 and 3 million years ago. This has become a major question in our evolutionary history, and I'll explain why. And if there was diversity, how many of them were there? Can we answer this question based on the discoveries from the Woranso-Mille? So you also talk about how did this related hominin taxa manage to live in the same area at the same time? Because usually we're told, when we're in school, we're told like this, there is something called competitive exclusion. 2 species cannot live in the same area doing the same thing. One has to go extinct. But if they manage to live together in the same area, probably at the same time, then we have to figure out how they manage to do it. And most of the information to answer this question comes from our understanding of the paleoecology in which these early human ancestors lived. And we think that we can extract a lot of data from the fossils that we're finding from the Woranso-Mille. But all of this information has to come from some sort of data. And our major source of data are the fossils, right? So when you do your data collection, you have to go to the field and find fossils. In other words, you have to do field work. So field work is the major part of our data collection. And when we go to the field, there are lots of preparations that we have to make, but I don't have time to talk about those preparations. But I just want to take you to the field right immediately and show you this is what we mostly do every day. We walk and looking for fossils and staring at the ground. So sometimes we look at fossils crawling like what you're seeing here, and you find fossils once in a blue moon, right? So this is a good discovery. That's a mandible. That's a jaw, half jaw of a hominin. But I just want to tell you the history behind fossil discovery. Now, you see that year, 2006? This is a specimen that we found in 2006. Sometimes we say, well, in order to find fossils, you have to be at the right place at the right time, which is true. Now, when we found this specimen, we always do surface scraping, sieving, and also picking and try to retrieve as many pieces as we can. But that year, we did a lot of sieving, but we did not find the other half. Now this is 2018. 2018, we find a small jaw that was really interesting, and we did the regular thing like sieving, picking. We didn't find anything else, but then I realized it looks like something that we found a long time ago, right? And we go back to the lab, and that's the one from 2006, and this is the one we found in 2018. 12 years later, we found the other half, right? because we were at the right place at the right time. But from a totally unexpected place that we found. So this is how we collect these fossils from the field, and sometimes daunting, sometimes hard. But when you find things like this, it becomes really exciting. So for the data collection part, I'll leave you there and see if you have any questions on how we do our field research.
So you told us the time difference between you found those two pieces. What about the distance?
The distance was 75 meters away from the initial discovery. But what was really interesting was when you look for additional fragments of a specimen that you find, you look at the modern topography and try to follow gullies. So that's what we did in 2006. We followed the gully that we could see and the immediate area, but where we found this one was on a totally different area. So I was asking myself, is this because the old channel changed somehow and what we had was like a new channel? So it was probably taken away in a channel that is now flat surface. Okay, that's one of my assumptions. But the other one is maybe just an Afar kid herding his goats probably picked it up and threw it on his goats. That's another possibility, because this happens a lot in the in the Afar. Kids just pick it up like a rock because they don't know it's fossil or a rock, and just they throw it on their goats. Find it, but sometimes, like this time, we were lucky to find that.
11,000 fossils. Can you just take them? Does the Ethiopian government one time would want them back as a treasure for their country? And the second, this is an open field, and you said that shepherds can take and throw things. What about scavengers that would come and either destroy or take?
To answer your first question, all those 11,600 fossil specimens, they never left the country. They never leave the country unless we have specific demands in terms of analyses. We don't take them out of the country. They stay in Ethiopia, in the National Museum, and that's where we go to study them. Okay. But there are some cases when, for example, we have like a specimen that we want to CT scan. And if they don't have the facility there, they allow us to export it abroad and get it scanned and return it for a short time. The second question, scavengers. Now, those animals have been dead for 3.6 million years. But when they were alive, there were scavengers. So scavengers must have been moving pieces and parts of those skeletons here and there. Some of them don't even get preserved, right? Some of them, the ones that are really lucky to be fossilized and found by us, would be preserved. So there are like rare occasions where you can actually find like a whole skeleton. It's really impossible to find a whole skeleton. I have never seen any, a whole skeleton found in the fossil record. Maybe dinosaurs, but not human ancestors. The most complete one is probably 50% or 40%, like Lucy. But those are like special ways of preservation. The animal must have died and got buried really quickly before it got scavenged or something. But scavengers have played a major role in terms of the distribution of the fossils that we're finding today.
Thank you for joining us. You've been watching Dr. Johannes Haile Selassie discussing the Woranso-Mille paleontological project in the Ethiopian Rift Valley and the diversity of fossil fauna, especially early hominins, found there. For more information on the Origins Science Scholars Program, please visit the Institute's website at origins.case.edu. In the next part of the talk, Dr. Haile Selassie will focus on the major hominin fossils discovered at the Woranso-Mille site. Now, back to the talk.
I gave you an introduction to how we find the fossils. Now, let's go to the fossils themselves and talk about some of the major discoveries from Woranso-Mille. Some of them you've already heard about. For example, Kadanumu. This is the partial skeleton, the 3.6 million year old partial skeleton that we found in 2005 and published it in 2010. The other one is the Burtele foot, which is also a very interesting specimen, and that has created a lot of controversy in terms of how we understand Middle Pliocene hominid diversity. The third one, which is also another point of controversy, a new species that we named from Woranso-Mille, known as Australopithecus deyiremeda. Now, these three specimens were picked up because each of these gives us or addresses the key questions that I brought up earlier. Now, first, I'll talk about Cadanomo, which is assigned to the genus, the species Australopithecus afarensis, which is Lucy's species, which lived from about 3 million years ago to 3.7, 3.8 million years ago. Now, remember when I said the Australopithecus anamensis, Australopithecus afarensis chronospecies lineage. Were they really ancestor descendant? And I also told you that there was that time gap where we didn't have fossils from, the 3.6 to 3.8 million years gap. Now, Cadanomo and all the other specimens that you're seeing here are from that time period. And they give us some information to test the hypothesis of ancestor-descendant relationship between Australopithecus afarensis and Australopithecus anamensis. Now, Cadanomo, the partial skeleton, its morphology is very similar to the skeletons of Australopithecus afarensis that we know of. So we don't have any problems assigning that specimen to Australopithecus afarensis. But there are also others, like the jaws that you're seeing here, which had a mix of traits, some of which are like Australopithecus afarensis, some of which are like Australopithecus anamensis. So those are like, they share morphological features with both the putative ancestor Australopithecus anamensis and its descendant Australopithecus afarensis. So we use those specimens to test that hypothesis of the chronospecies lineage of those hominins, Australopithecus anamensis and Australopithecus afarensis. So what we said was, we cannot really say that these specimens, except for the partial skeleton, are either Australopithecus afarensis or Australopithecus anamensis, because they share features with both. But they really represent good evidence to show that the Australopithecus afarensis, Australopithecus anamensis lineage was actually an ancestor-descendant lineage. So that question gets tested by what we have from here. And that's where I'm gonna stop answering that question. But the second and most important question is about diversity. Now, hominin diversity, particularly for the time between 3 and 4 million years ago, was never a question, because there was none until 2001 or 2002. Because everything that we knew before 2001, as the earliest human ancestor was Lucy's species, Australopithecus afarensis. But people knew that after 2.5 million years ago, there were multiple hominin species living at the same time, based on fossil evidence from Ethiopia, from Kenya, and some other places. So if you look at the pre-2010 Pliocene hominin diversity, what you're seeing here is Ardipithecus ramidus at the bottom became the oldest species at 4.4 million years ago. Then you have Australopithecus anamensis, which is the putative ancestor of this lineage, Australopithecus afarensis. One species was named from Chad in Central Africa, which is known as Australopithecus bahal-ghazali. And another species was named from Kenya called Kenyanthropus platyops. Now, this has been known since the 1920s, particularly Australopithecus afarensis has been known since 1924. And what you're seeing here is the robust lineage, Paranthropus robustus, also known from South Africa since the 1930s. So this was what we knew in around 2010. So people started talking about, well, maybe we have some kind of diversity at about 3.5 to 4 million years ago. And what you're seeing here, repeating the slide in a different way, is that there are multiple taxa living during the Middle Pliocene, meaning Australopithecus bahal-ghazali, Australopithecus afarensis, and Kenyanthropus platyops coexisted. This is what appeared to be the case when people named those two taxa, in addition to the well-known Australopithecus afarensis. Of course, this was not accepted by every paleoanthropologist, particularly for those who've been arguing for like 40 years that Australopithecus afarensis is the earliest ancestor And it gave rise to everything that came after that. So the argument is that we're just looking at Australopithecus afarensis. There's no Bahr el-Ghazali, there is no Kenanthropus platypus. These are just geographic variants of the well-known species Australopithecus afarensis. And they talk about the differences being just superficial, like having all of them wearing caps or glasses, right? So this is what a lot of people think about Australopithecus Bahr el-Ghazali and Kenanthropus platypus. Now, you find a foot like this, then it becomes a problem to put it into Australopithecus afarensis, because this foot is not a foot of Australopithecus afarensis. It's a totally different foot, and you can see why. It has opposable big toe. And if you've heard about Ardipithecus ramidus at 4.4 million years ago, that's also the species that has opposable big toe. Now, is this related to Ardipithecus ramidus? It could be. There is a difference of 1 million years between this and Ardipithecus ramidus. Could they be related? Of course. But are they related? That's something that we have to test, not only based on the foot, but also other craniodental material that we haven't recovered yet from this locality. So the Burtele partial foot became one of the convincing elements in terms of showing diversity during the Middle Pliocene. So we're talking about two different hominins adapted to two different modes of locomotion, bipedal locomotion. Because Lucy's species, Australopithecus afarensis, as most of us know, was bipedal. And there's not only morphological evidence for that, there's also evidence in the form of footprints from Laetoli in Tanzania that are dated to about 3.6 million years ago. Those footprints clearly show that Australopithecus afarensis was bipedal. Now, you cannot make this footprint if you have that Burtele foot. You're going to have a totally different footprint. So there was no doubt about Burtele foot representing a species other than Australopithecus afarensis. So it's more like Ardipithecus ramidus, the early Ardipithecus ramidus, which also has opposable big toe. Okay, so looking at those two, superficially, yeah, they look very similar. But when you look at the detailed morphology of the feet, they have a lot of differences. Not only size, but also detailed morphological features of the joint surfaces and so forth. And we refrained from putting it in the Ardipithecus because we don't have enough morphology to actually convince ourselves to put this into Ardipithecus. So that became one of the strongest evidence to show that there were at least two different species living at the same time in the Afar region of Ethiopia 3.4, 3.5 million years ago. Then after a few years, we found more specimens from various localities, and we named a new species known as Australopithecus deyiremeda. Now, let me just give you an idea in terms of the distribution of these fossils on the landscape. Here is Kadanumu. This is the partial skeleton that we assigned to Australopithecus afarensis. Here is the Burtele partial foot. Noforaitu is another locality where we have remains of Australopithecus afarensis. Right here, close to where we found the Burtele foot, is where we found this new species of Australopithecus deyiremeda. It started with a discovery of few specimens that you see on this slide. A partial maxilla with some dentition, a edentulous jaw, a fragment of a maxilla, the upper jaw, and another lower jaw fragment, of which we found the other half and became really a beautiful specimen. And this was one of the paratypes of the species that we named as Australopithecus deyiremeda. So most of the morphological features that we use to distinguish this from Australopithecus afarensis, Lucy's species, was based on the morphology of these two specimens, the upper jaw and the lower jaw. And we came up with characters that to us seemed different from what we see in Australopithecus afarensis and Kenanthropus platypus. Unfortunately, we couldn't compare it with the other species from Chad because the Chad species is represented by lower jaw fragment, and it doesn't really correspond to the morphological features that we see in this specimens. So we talked about the size of the teeth and detailed morphology of the teeth. We also talked about the shape of the canine, which to us looked very different from what we see in Australopithecus afarensis. This seemed to be more primitive than what we see in Australopithecus afarensis. We also looked at enamel thickness of the molars of this specimen, and it looked to us that this is thicker enameled than Australopithecus afarensis. So the combination of all this, particularly the robusticity of the jaw, the lower jaw, tells us that this is something different from what we know of Australopithecus afarensis. Now, when we think about Australopithecus afarensis, it is one of the most dimorphic species that we know in the fossil record. So if we can exceed that dimorphism by what we're seeing here, then it's only reasonable to assume that this is a different species. You can't simply keep adding differences to that species and just inflate those differences artificially as if they belong to a single species. So I don't expect you to read this, but I just want to show you that some of the features that we saw as differences between Australopithecus afarensis and Australopithecus Kenanthropus platypus and Australopithecus barghazali were really significant. They're all related to dentition and corpus or jaw morphology. So we were able to identify some differences between afarensis and Kenanthropus platypus, and also between Australopithecus afarensis and Australopithecus deyiremeda. So all these differences were convincing enough to us, at least, not to our colleagues maybe, that we were dealing with a new species. So we named a new species Australopithecus deyiremeda, and we published in Nature in 2015. So when you think about why are people really doubtful about this new species that we are naming from this specific time period? Now think about Australopithecus afarensis. Afarensis has so many specimens known in that group of species, and the kind of dimorphism, the kind of variation that we've documented in that species are so enormous, in some ways even exceeding the variation that we see in some modern taxa. And if that can accommodate whatever we're finding, then you would probably agree that this is probably just one species that lived between 3.2 and 3.8 million years ago. But that was not the case for us. And so with all these discoveries, we had to actually rethink about all these phylogenetic relationships that we've been learning in school and following for many years and teaching our students the same thing. So the idea before 2000, 2001 was that there was this lineage, with Australopithecus afarensis, of course, being the oldest ancestor, giving rise to the lineage that led to humans. So that's the Homo lineage. But there was another lineage, which is the South African hominin lineage, including the Paranthropus species. So it was as simple as afarensis being the stem group that eventually gave rise to both the Homo lineage and the Paranthropus lineage. So at this point, we know, at least from about 2.5 to 2 million years ago, there were multiple species living at the same time. So nobody doubted this, because we had Homo and Paranthropus overlapping both in Eastern Africa and Southern Africa. So here you have a good evidence for overlap of related hominid species living at the same time, right? But we never had that evidence for the time before 2.5 million years ago. But now, that tree that you saw on the previous slide has changed so much. We have almost doubled the number of species that we know in our fossil record, right? So what that means is that you see two time periods where there was diversity of these early human ancestors. Now, a lot of our colleagues would argue that Australopithecus barghazali, Australopithecus deyiremeda, and Kenanthropus platypus are all geographic variants of Australopithecus afarensis. They would prefer to put all of it into a single species. But as you can see, the Burtalephid is not included in that oval, right? Because no matter what you do, that foot is not going to belong into this group as Australopithecus afarensis. So you have good evidence of at least the presence of at least 2 early human ancestor species between 3.3 and 3.5 million years ago. Questions?
Africa is a huge continent. Are other groups finding other evidence elsewhere, or is it all limited to this part of that continent?
If you're talking about this specific time period between 3 and 4 million years ago,
Anytime.
anytime we do have evidence of early human ancestors from different parts of Europe, China, Africa, but if you're talking about early human ancestors before, for example, like 1.8 million years ago, all the record that we have is only in Africa. But at 1.8, we have a record of a species known as Homo erectus from Georgia, the country, from a place called Dominici. We have Homo erectus remains from Indonesia at 1.8, 1.7 million years ago. We have Homo erectus from China, about 500,000 years, or probably goes as early as 1 million years. So there is evidence of early human ancestors living in different parts of the world after 1.8 million years ago. But before that, all of the evidence comes specifically from Africa.
I'm interested in the logistics of how you put together your field research team.
Am I allowed to spend an hour to answer that question? Well, field logistics is really difficult, to say the least. We not only think about the personnel that goes into this effort, meaning who should be involved as a personnel, as a scientific personnel, who should be involved as logistics personnel, who should be involved as like local personnel and so forth. So there's a lot of planning that goes into, even before getting into the field, you have to do a lot of preparations. But once you're ready, you're all set, then you have to find grants to do your field research, which is not easy. You have to, particularly in our case, the afar is just in the middle of nowhere. So there are no like convenience stores or anything, or restaurants for that matter. And we have to buy everything that we need for like 6 weeks. We have to buy it all in Addis, and we have to transport everything that we need, including tissue papers, and set up camp in the middle of nowhere and stay there for like 6 weeks in 110 degrees temperature. So it's really demanding a process in terms of organizing a field team all the way to going out to the field, and the time you spend before you even start looking for fossils is so enormous, both in terms of manpower and also financially. But in the end, when you find fossils, you don't feel all that just like cool.
Looking at the human family tree, do you think all of that was the result of one common ancestor and adaptive radiation, or because you had 2 separate bunches, is it possible each had a separate progenitor? And the other question, if you don't mind, how do you date a footprint?
All of the hominins that you saw on that family tree, they had one ancestor deep in the past. But once you get to like 3 million years, 4 million years, and you see diversity, that is one of the things that we could argue that this could be adaptive radiation. Now, one of the major research questions that we have right now is, because we have multiple taxa living in the same area at the same time, we're questioning how did they manage to live at the same time and place together? Were they like partitioning the habitats? Were they living in different places but close to each other, or were they exploiting like different food resources? Those are the kinds of things that become really interesting, and that's the subject matter of what I'm going to talk about next. So I'll give you more answers on that. And the second question was,
How do you date a footprint?
how do we date a footprint? Good question. Fortunately, the footprints at Laetoli were on a volcanic tuff, which is usually what we date and give us like dates for the fossils that we're finding, because normally we don't date the fossils directly. What we did are the volcanic tuffs above and below this fossils. So those footprints were actually on a volcanic tuff, which was then dated to about 3.6 million years ago. But if it were like on clay, then you're going to have to hope that there is a tuff above it and below it to have a date for that. But that was phenomenal that it was on tuff. So it was a direct date.
We hope you've been enjoying the Origin Science Scholars Program with Dr. Johannes Haile Selassie. Dr. Haile Selassie is curator and head of physical anthropology at the Cleveland Museum of Natural History and a fellow of the Institute for the Science of Origins. In the second part of our talk, we learned about great Woranso-Mille finds like Karanumu, an Australopithecus afarensis skeleton over 300,000 years older than the famous Lucy, Australopithecus deyiremeda, a new hominin species, and the Bertelle foot, which suggests that there was more than one hominin species living in Ethiopia at the time of Lucy. In our final segment, Dr. Haile Selassie will talk about how scientists reconstruct the habitat of sites like Woranso-Mille millions of years ago. Now, back to our talk.
Just to start from his question, we talk about many species living at the same time in the same area. So how do they manage to live together, right? He mentioned adaptive radiation, for example. That could be one way. But what causes adaptive radiation, right? And that brings us to understanding the need for, to understand the paleoecology in which these early human ancestors lived. So my next talk is going to be just on paleohabitat reconstructions and how do we do like habitat reconstruction to reconstruct an environment from 3 million years ago, 4 million years ago is not going to be easy, right? But scientists through the years have developed many techniques in terms of addressing paleoenvironmental reconstruction. So they use methods like community structure analysis, isotope analysis, and something we call ecomorphology, trying to look at the shape of the bones and how they relate to function, and among so many other methods that people are using. So when you look at Lucy, for example, this is her partial skeleton. As paleoanthropologists, we look at the fossil bones, each element of her body. We try to understand the morphology, try to understand her locomotor adaptation, the size of her brain, and so forth. And with the help of paleoartists, we give her some flesh, right? So maybe this is what she looked like. But where was she living? Where was she actually living? Was she living like in a wooded habitat like what you're seeing here? Or was she living like in more open habitats? So how do we find out? We need the answer, because if we're going to talk about adaptive radiation, we have to know where they were living, right? Were they living at the same place, or were they living at different places in the same area, and so forth? So we try to reconstruct this past habitats and past environments, first by trying to understand the extant forms, the living forms, because the assumption is that these living forms look like their ancestors in the past. So whatever they're doing, their ancestors must have done similar things, right? So some animals have very specific habitats that we call indicator species, and we try to see if we have those indicator species in the fossil record and try to reconstruct the past environment based on what we know from the present, right? And people do all kinds of analyses in terms of community structure, and what we have done with Dr. Su is look at the community structure using what's called correspondent analysis, and we look at the localities that we have from the Woranso-Mille, and what you're seeing here is that the Woranso-Mille localities, which are in this black squares that you see here, are distributed here, and what that tells us is that most of the localities fall into the area where we have like woodlands, bushlands, and some open grasslands, and they're not different from all the other hominin-bearing localities like Tanzania, like Toli, Tanzania, or Hadar in Ethiopia. So these hominins were all over the place in terms of their distribution, and they were probably exploiting diverse resources. They were not really limited to anything. So in addition to the correspondent analysis, we can actually look into the details of their teeth and try to register the isotope composition of their teeth, because when whatever plants they eat will have to be like either C3 or C4, meaning grasses or leaves, they're browsers or grazers, and the teeth actually keep the signal from what they were eating. So C4 plants usually indicate like grazing, and C3 plants indicate browsing. So people have done a lot of research, sampled early hominin teeth, and looked at the isotope contents of this teeth. And this is one of the works by a colleague of ours, Sponheimer. And what he has found is like most of the hominins that you see here have very diverse dietary resources. Some of them are limited to, for example, like C3, like what you're seeing here in terms of Australopithecus anamensis and Australopithecus sediba, and also Ardipithecus ramidus. But the others, you can see that they are distributed across a wide range of resources. And when you come here, particularly the Boisei lineage, they're C4. They use C4 food resources. So obviously you can see that there is some difference. But for most of the hominins that we have in the middle, they're all over the map, right? So we did the same thing. We looked at the Woranso-Mille hominins. We sampled about 14 teeth from Woranso-Mille and tried to look at the isotope. And what we're seeing is they're also distributed across, like mostly in the negative, but some of them seem to be like more to the open side of the habitats. But if you combine the whole data set, what you see is that Woranso-Mille hominins are like all the other hominins that we know. They're just consuming whatever food resources they can get their hands on. So people have been arguing about hominin habitat as mosaic. Hominids were able to adapt to various environments at different times, and they were not really specific in terms of their food resources. What that means is they could actually live in all kinds of environments because that's an advantage for them. They can be really specific, and if you think about those that are really specific about their dietary habits, they did not make it. They went extinct when probably the environment changed. Okay. But if you can adapt to various types of environments, then the chances of survival are much higher. So what we're seeing here in terms of the Woranso-Mille habitat is like all the other hominids that we have from other localities. They were living in a diverse setting, which included closed woodland, bushland, open woodland, shrubland, grassland. So they could exploit whatever was available for them. Now, the problem is when you have two species living at the same time in the same place, did they really exploit everything that was there without any problems? Or did they actually had some niche partitioning, like they were excluding each other from overlapping? So that's one of the questions that we have right now in terms of addressing the paleohabitat of Australopithecus afarensis and Australopithecus deyiremeda, and also whatever species the Burtele foot belongs to. How did they manage to live in the same area at the same time? So that's one of the major research questions that we're trying to address now. But overall, when you look at all the evidence that has come out of the Woranso-Mille, you realize that this is one of the best places, if you will, particularly for the time between 3.5 and 4 million years ago, to address so many questions in human evolutionary studies. So to summarize the major significances of this Woranso-Mille site, one, it samples a time period that's poorly known in the fossil record, particularly the time between 3.6 and 3.8 million years ago. Secondly, you've seen that there are about 11,600 specimens that have been collected from the place. And even though it's like 2%, it's a good number of hominins represented at this site. But most importantly, it gives us a lot of information in terms of testing the hypothesis of ancestor-descendant relationship between Australopithecus afarensis and Australopithecus anamensis. And finally, Woranso-Mille is probably the only site that has yielded the best evidence for the presence of at least 2 hominin species during the Middle Pliocene, particularly around 3.3 and to 3.5 million years ago. So those are like major points that we were able to address based on the discoveries from Woranso-Mille.
I don't want to minimize the drama of finding many species present at the same point, but I do want to question the conventional older wisdom that you couldn't have 2 species in the same ecological niche. When you look around, you might find, I mean, even today, you can see grain-eating songbirds in Cleveland as a niche, and there's a very large number of species in that niche. And I'm wondering what's the difference between the conventional point of view and that observation and your observation on multiple fossils at the same time. And as far as I can tell, the conventional view assumes that they would be competing for resources like food, and if one was slightly more successful and the other could drive it out. But there's an unexpressed assumption in that, and that is that the pressure on the food supply comes up to saturation of the food supply. If the food supply is much greater than the requirements of the 2 species, why in fact should they compete to the exclusion of one another? And that's the question. Can you explain the conventional wisdom on species not existing at the same time in the same niche?
I think the conventional wisdom should be open for testing. We can't just take it for granted because it's conventional wisdom. But what we're seeing even in the modern world, we have so many taxa that are related, closely related, but living in the same area, doing the same thing. Now, what that means is that competitive exclusion is not at work because the resources are abundant, that they don't have to compete for those resources, right? So that's exactly what we're trying to test here. If we're seeing evidence for the presence of 2 species, is it because there was enough resource that they didn't have to go through the competitive exclusion? Or is it because they're doing 2 different things? For example, one of them could be more into the closed habitat, and the other one could be to the more open area. Because what we're seeing in the Woranso-Mille area as other places is that no locality shows entirely forest or entirely open grassland habitat. What we're seeing is a mosaic of habitats that are present adjacent to each other. Okay, so you can have 2 species doing different things, but few miles apart from each other, right? So we cannot really say, well, the wisdom, the conventional wisdom is competitive exclusion. We can't really rule out the possibilities of 2 related taxa living at the same time in the same area. So that's something that we're trying to test. And all of the data that we're collecting, all of the methods that we're developing is to help us address those kinds of questions, because we know a lot about so many conventional wisdoms. But as far as I'm concerned, each conventional wisdom has to be tested. And if it remains a wisdom, we'll take it. If not, it should be falsified. So what we're doing is exactly that. We know the conventional wisdom, but we're trying to falsify that.
You said that the same species could adapt to different habitats, and I was just wondering, as an engineer, how they would do that. Would they put on clothes and take off clothes with the temperature, or have different tools or weapons for hunting or versus gathering plants to eat, or how could they, what was the mechanism whereby they were able to adapt to different habitats?
It's mostly being able to consume different food resources from wherever they are. So it's more about the food resources than where they actually living physically. Because they can move from place to place looking for those food resources. Some places they might stay for a while because there are plenty of food resources. At other times they can move over somewhere else. But those places that they go to could sample different habitats. Here it was probably, they were probably eating a lot of fruit, and it could be like a little, woodland. But when it's over, they might go out to the more open area looking for something else. So the fact that they were able to exploit those different habitats is what is meant by, they were able to exploit, different places, yeah. And with that, thank you very much.
The Origin Science Scholars Lectures are presented by Case Western Reserve University's Institute for the Science of Origins, with the assistance of the Siegel Lifelong Learning Program, the College of Arts and Sciences, and MediaVision. For more information on the Origin Science Scholars Program, including a full video archive, please visit the Institute's website at origins.case.edu.